And everyone, welcome to session 8 for today. Too close. Welcome to session 8 for today, which is on invasive species. And can I please just remind people in the audience to please scan and adjudicate this session on page 12 in your program. You can scan the code and then you can judge and adjudicate the presentations that will be presented in this session. And to bless us today, or to open up our session today, we have Monica Ndlovu, who will be presenting on assessing the spread of an emerging weed in KZN. Is Monica? Yes, she's here.
Okay. Good day, ladies and gentlemen. My name is Monica Ndlovu, and I work as a research assistant under the eThekwini Municipalities Biodiversity Management Department. So today I'll be sharing just a short mini study that we did on our project site titled Assessing the Spread of Porophyllum ruderale as an Emerging Weed at the Buffelsdraai Forest. So it was 2023 when I first noticed a plant that was growing quite a lot around our offices and along our roadsides. I then tried to identify the plant. I couldn't. I then uploaded an image of this plant on iNaturalist, and that's where I discovered that it was an exotic that was not commonly observed within the eThekwini area. So here, I've just included a picture of the plant. So Porophyllum ruderale is commonly known as Yerba porosa, onica, or Yerba Del in its native area. The Porophyllum genus falls under the acerosum family, and this plant is believed to be an annual herb or shrub, and its native range is the North and South America. And in one of the countries within its native range, Mexico, 17 of the Porophyllum species are found there. 17 of 25 of the Porophyllum species are present in that area. And Porophyllum was listed as one of the most, Porophyllum ruderale, to be specific, was listed as one of the most distributed within Mexico. So Porophyllum has opposite and alternate leaves that are about 6 to 25 millimeters long. The leaves are ovally shaped and they are wavy at the edge. It also produces numerous flowers that are considered to be hermaphrodites, and they are found to be greenish, purplish, with tubular, and they are long and thin, and they have curved branches. And the plant has been seen to grow to a height of from 30 to 60 centimeters, and sometimes it can be found at a height that is over a meter. So normally the plant flowers between August to October, and it produces achenes that are about 9.5 to 12.4 millimeters long. And it has been typically observed growing in a variety of soil types. And it has, in its native area, it has been found that it grows a lot in low, flat areas, such as agricultural fields. And the type of fields that I saw, or that I read that this plant is found in are like normally crop maize, sugarcane fields, and sometimes rice fields. And just like other exotics, it grows a lot in disturbed areas and roadsides. And because of the type of seed that the plant produces, it's spread quite easily through wind, water, wildlife, and humans. So with regards to its uses, I just included an abstract that I saw in one of the papers that was studying the plant's medicinal uses, but the plant itself, it's considered to be edible, and it's used as a seasoning in most Latin American cuisines because it is believed to have a similar taste to coriander. It's also consumed by animals, mostly cattle, and some wild animals. I think an example that was provided for Mexico was, I think, the white deer. And then as here we have the medicinal properties of Porophyllum, and they include anti-inflammatory and antimicrobial and wound healing properties. And some components of the plant are used in treating digestive issues, liver, kidney disorders, hypertension, and snake bites. So currently, I could not find when the first record of Porophyllum was identified within South Africa, and that then means that the plant's negative impacts are still, well, they're understudied within the country, but in areas where the plant has been identified outside its native range, there are a few negative impacts that were observed. And some of these are it's the ability of the plant to become like very weedy. It's a weed. And some people don't like the smell of the plant, especially if the leaves of the plants are crushed. And in other places, it's listed as an invasive. So because it's understudied in the country, I could say, well, it's not listed as an invasive currently, but we then decided to observe and monitor the species after we observed or recorded the first observation of this plant within our site in, So what I found was, when I uploaded the image on iNaturalist, I saw that there was an existing upload of the plant. And that image was uploaded on the 9th of March in 2022. So the objectives of our study were to assess the plant's density within the study site, and also try to identify how the plant is distributed within the study sites or our project sites management blocks, and also understand which blocks are likely to be encroached by the plant. So here, this is our study site as shown in the map. The Buffelsdraai landfill site is located 25 kilometers north of Durban in KwaZulu-Natal. So the area that is in the middle, or the footprint of the landfill, is shown in the middle, and it's marked by 0. And the surrounding area of the landfill forms the Buffelsdraai Forest, which is the buffer zone to the landfill. So that Buffelsdraai Forest is then divided into 30 management blocks, and these blocks make up, they are about 800 hectares in size in total. And historically, the whole site was used for sugarcane farming, including the landfill. But the municipality bought the site, and the biodiversity management is currently managing the buffer zone, or the Buffelsdraai Forest, under their community reforestation program. So in order to carry out our study, we then decided to do our field observations between October 2023 and March 2024. That's where we normally saw that the plant had flowered around then. We then checked for the presence or the absence of the plant through the site, through field observations. And then we then decided to do random sampling on pre-selected plots within all the management blocks. There, we then measured an area of 30 meters by 30 meters, making up a sampling plot. And within the sampling plot, we used a quadrant that was about 0.5 by 0.5 meters that was thrown around the sampling plot 10 times. And all of the Porophyllum plants that were found in the quadrant during the time that we were sampling were counted, and then we then calculated like general density of the plants within that plot. So once we had done with our calculations, we then went out to collect the specimens of the plant in different parts of the project site, and those specimens were then pressed and submitted to the herbarium. And then we then took images of the plant and different points of the site, and those georeferenced images were uploaded onto iNaturalist. Okay, because of the time, I'm not going to go into what iNaturalist is. I think we also Savannah's presentation yesterday, but here I just wanted to provide like an overview of the global distribution of the plant on iNaturalist, since that's where we will be mapping our plant. And here, as we can see, it's quietly populated within its native range, but it is seen in other areas outside of its native range, and including South Africa. And in those areas that the plant has been seen outside of its native range, and other places it has been listed as an invasive, such as in Indonesia, they've listed it. So here I just provided two maps also, like showing an in-depth view of the distribution of the plants in the year 2021. We just wanted to see that how populated or how spread it was around our project site. And as you can see, the first picture was from observations seen in the year 2021. And then currently, that's the spread of the plants within eThekwini. So for our results, we then saw that the plant is actually present in all management blocks, 90% of the management blocks actually, and that the mean density across the sampling plots was 0.48, and it actually had a high variability. So as much as it was seen across the plants, the densities were they were inconsistent. So yeah, and then some of the plots showed high concentrations, and then while in others, the species was absent. And then from the concentrations that were calculated, we then observed that the plots that are likely to be invaded or encroached in the near future are block 1, 0, 10, and 15. So here I then did like a map showing all of the points where Porophyllum had been observed on site. So these are some of the points that I extracted from iNaturalist. Most of the observations were done by me, some were then done by my colleagues on site. And then the red points on the map show the points we collected specimens of the plant. So within those points, it was just not just one plant, but it had a population of the plant. And then this is the boundary of the site. The site itself doesn't have a physical boundary, it's not fenced, we just have trees. So in conclusion, we saw that Porophyllum has naturalized within the project site. This then calls for like future research for us to understand how it impacts the environment as it grows in, and we could then probably do further research and understanding the control methods on how to control the plant. And then from this study, we decided to create a project page on iNaturalist to monitor the growth of this plant within eThekwini. So here I just included a picture. So this picture was a picture that I took in block 10 in December 2024. I just wanted to like give people an idea of how the plant grows or how it encroaches an area. So here we had just done brush cutting to open pots inside the block. So this is not a roadside actually. And this is how the plants, all the seedlings in between show are mostly Porophyllum. So it has like a mat forming habit when it grows, especially in a disturbed area. Yeah, thank you.
Thank you, Monica, for a lovely presentation. Unfortunately, we don't have enough time for questions for you, but I'm hoping that at the end of the session, there will be some time and then we can do a discussion. But we can welcome our next speaker now, Michelle, Keith. to the stage where she will be talking on a presentation on the evaluation of two herbicides as a control method, sorry, yes, for an indigenous encroacher in, I'm off today, in KZN also.
Good afternoon, ladies and gentlemen. It's great to be here at the 60th Congress, and quite an honor to present these results, which have been a long time coming, unfortunately. So what do? We have an indigenous plant species. It's relatively unknown. It's encroaching. Limited research has been conducted, and it's thickening at a rapid rate. It's having an ecological and economic impact. And effective management solutions haven't been found as yet, and they haven't been quantified. The time taken to research possible solutions are costly, both economically and ecologically. If we look at Pseudomonas plumosum, for instance, it was identified as a problem in the 1950s, and it was only in the 1990s and 2000s that we started seeing solutions coming forward for our farmers. So in this case, what do? How do we eat this elephant of the unknown? Well, one small bite or one small step at a time. And where we start these small focused steps is often identified by those most affected by the encroacher. And as a researcher, we want to present a viable solution to prevent desperate measures being used in an attempt to control the plant. So I'm looking at evaluation of the two herbicides to control Phymaspermum. And here the beast is, pretty plant, flowers in April. It's indigenous, as I keep repeating. It's multi-stemmed, it's densely leafy. It's widely distributed naturally in South Africa from PE all the way up into the Limpopo province. And it occurs naturally in small colonies and grasslands and in the forest margins. It has similar life traits to other indigenous encroacher species and alien invasive species, in that it has a rapid growth form and it resprouts readily from a thick woody rootstock. It has high seed production, and dispersal is easy as it has small seed size. It's tolerant to fire, highly unpalatable, and very environmentally adaptable. As I say, it's encroached into our grasslands, and it's coming to the forefront more and more with different people in the areas. It's impacting both our livestock and felt production, which is impacting food security, biodiversity, and farm profitability. If we look at encroachment, and this is meant to be a video, I'm not too sure how to get the video activated. If you can, please, I'm not too sure. Nope, okay. So the known factors are grasslands are ecologically sensitive. Fire doesn't kill Phymaspermum, but it does cause top kill if we have enough biomass. And this induces rapid regrowth from the roots in spring. We have no registered herbicides for controlling it as yet, and the association between Phymaspermum and other alien invasive plants is well known in the area. Landowners are requiring a solution which is both cost effective to them and environmentally sensitive. So what is unknown? Our herbicides, which type, what dosage, what application method, and when do we apply them? So our objective is to identify a cost-effective selective herbicide. We don't want this. We don't want the napalm effect of killing everything as we go along. That's definitely not what we're wanting. So our trial site's located on Boschberg Nature Reserve in KZN, about 9 kilometers from Boston. And as you look at the Google Earth image, we can see that area there is a solid mass of bracken fern. And in both our trials, we had quite a lot of American bramble in this area. And in trial 2, we had a lot of American bramble. All those green dots in these areas is all Phymaspermum plants. That's the forest. So our trial sites, between 1,500 meters altitude, gentle slopes, the mean annual rainfall long term was 855 mils, but during our trial period, this was a lot higher. Our mean temperature range was between 8 and 20. Our soil form was a magma 1 to 100 of varying soil depths. It was in the Drakensberg foothill west grasslands. Trial 1 was southeast facing, trial 2 southwest. Both trials were within the same camp and had the same management applied to it. And we excluded cattle for 28 days post-treatment. We measured many plants. At the beginning and the end, we looked at canopy volume, we looked at basal area and the number of stems. And then on a monthly basis, we checked for survival. We measured plant heights, the number of stems, and the basic phenology. Our first treatment was to burn everything in winter and early spring. Our experimental units or replicates were individual plants, which were healthy, above 20 centimeters in height, and not seedlings, and most importantly, more than 2 meters apart. The choice of herbicides was difficult. We needed to think about the environment, this being a nature reserve, and also we needed to know a basic toxicology and long-term effects of the herbicides. So we looked towards what was being used on alien invasive species in the area, and that helped guide us to metsulfuron methyl, 600 grams wettable granules, and tricloper as an emulsifiable concentrate. We had 4 concentration rates of each herbicide, which we tried to get a dose response from, and 1 untreated control. So we had a randomized block design, and again, each plant's a replicate. So there were 44 plants per treatment in trial 1 and 48 in trial 2. It was just the number of plants available which we could use. And I was always told by statisticians, more is better than less. So I went with that. I applied the single foliar spot spray in November 2018 and 2020, standardizing all the equipment and the methodology. All herbicide treatments were applied to each block before we moved on to the next block. And we started with tricloper and every time because it is more volatile than metsulfuron methyl. We monitored climatic conditions closely and we stopped spraying if wind speeds were above 5 meters a second and temperatures were above 25, again, because of tricloper's volatility. So the jelly tots that we see, the jelly tots that we see there, or in some cases, licorice all sorts, are all individual treatments that were applied. So if we look at our plant size differences before the treatments, we know that this could have an effect on our herbicides if they are effective or not. And we can see that although there were no significant differences in the height of the plants, they were pretty uniform, but our basal area, our canopy and volume, and our number of stems, there were highly significant differences between the 2 trials, with trial 2 being substantially bigger plants from that perspective. And within the canopy volume of trial 2, we saw there were a slight significant difference in sizes. But this is where it just didn't matter. All that work. This plant is highly susceptible to herbicide control. We can only see a significant difference between untreated plants and treated plants. And you can see with an 86% mortality rate, we're good with that. We can take this and we can run. If we monitor the monthly survival regrowth, and we can see within the first 3 months, here, we saw a slight die-off. We saw the emergence within the first 3 months, I apologize. And then every now and then we would have plants dying off, and that was as a consequence of trampling or mold disturbance or animals digging. Anything went, the survivors died. Frost had no impact, and sadly most plants flowered, even though they were a little bit smaller. They all flowered within 6 months of spraying. So if we did see retarded growth of the survivors, it affected both the stem height and the stem numbers. And if we compare the two, the untreated and the treated, we can see there's a highly significant difference between the treated and untreated in terms of height. That is our critical value over there. So you can see the significance, how significantly different they are. If we look at the overall stem reduction, we can see there's a significant reduction again in stem numbers. Even in the untreated plants, we get natural self-thinning. But what the take-home message is, despite the 40 and 60% self-thinning that occurs in the untreated, we're getting 90% reduction in stem numbers. From a cost perspective, this always talks to everybody when you're trying to come up with money. We can see that metsulfuron methyl as a group is much more cost effective than triclopyr. And when you pair this up with the mean mortality rate of 96%, I'm starting to see where the best solution is. These are the most expensive herbicides that I could quote on. If we use generic, which are herbicides which are far cheaper, we're going to see a 40 to 60% further saving. I just want to add here, we excluded labor because it's highly variable as to what that cost is. So this is pure herbicide plus adjuvant mixture. So in conclusion, what can we say about this? Both herbicides as a single foliar application controls Phymaspermum in early stage of growth. Survivor regrowth is severely retarded and it impacts both seed production and dispersal. 1 gram metsulfuron methyl is most cost effective. And the association with bramble and bracken works in our favor because they have higher rates of application. I just want to point out all herbicide formulations in South Africa are toxic and all label recommendations are there not for a reason. They must be followed. The way forward, briefly, if I may, we need to still explore other herbicides, especially if they have lower impact and toxic effects and different modes of actions. We need to explore the treatments in different seasons by climatic areas on different plant sizes and the time since they were burnt. And furthermore, the better solution would be to explore management interventions or combinations thereof by identifying the drivers of the current spatial and temporal distribution, which should provide a guidance. I would like to thank everybody. There's a huge team behind me over the season, the seasons and many seasons. And I think I talk for everybody. We stand up here alone, and you probably find there are 30 or 40 people behind us, supporting us on this adventure. Thank you so much.
Thank you so much, Michelle. Unfortunately, there's no time for questions for Michelle, but you can grab her during tea afterwards. And now we get to invite our previous chair to the stage, Errol, who will be presenting on optimizing the frequency. Thank you. Who will be presenting on optimizing the frequency of mechanical control of the bracken fern. All yours.
Afternoon, everybody. I'm back. So, as you would have heard, I'm going to be talking a little bit about bracken fern and how do we optimize its control in some of our grassland areas. All right. So, I'm sure most of you know about bracken fern, and you probably also know that it encroaches into many of our natural grasslands. It is considered indigenous, and interestingly, in the research, the reading I've done, it seems to be indigenous to most parts of the world except for Antarctica. So, very widespread plant. But it unfortunately does appear to have this ability to transform grasslands and definitely encroaches. It seems to thrive when there's disturbance, including from fire. And in Durban, we have previously appointed working on fire as an implementing agent to look after our grasslands. And what a lot of people were saying is, don't burn the bracken because it just comes back stronger. But of course, we need to burn our grasslands because the fire stimulates germination of other species. So we said, well, what is the problem? Well, as I've said, we know it regenerates, and it's got these underground rhizomes which can spread. And you see I've plucked out some of the plants here. Those are not the rhizomes. You can see that's just the underground portion of the frond. The rhizomes stay underground unless you literally go and dig down and try and find them, which again is not something we're going to do in our natural grasslands. Cutting seems to stimulate regrowth. But it's all comes down to the timing of cutting. And obviously for many of you in grassland science, this will make complete sense. If you overgraze an area, what's going to happen? You're probably going to damage your grasses. Maybe the roots will start to become, depleted. And that's exactly what we decided we need to do with bracken because we don't want to use herbicide in our grasslands either. We've got very high diversity of forbs in the grasslands that we're trying to look after. So the study objectives, we said, okay, how do we go about looking for something that's improves our cost efficiency? And we decided on manual control. As I said, we don't want to use herbicide. We want to try and suppress bracken, but ultimately balance the effectiveness because of the operational costs. So because we've got working on fire and the teams going onto these sites to do alien plant control in the summer months and fire control in the winter months, and we've got a lot of sites. We've got about 50 sites we're managing in and around our municipality. So cost is a big factor. So we chose 6 of our grassland sites, and we decided, well, we'll put the teams in there every 4 weeks, every 8 weeks, or every 12 weeks to do some cutting. And then we'll go back and measure the density of the fronds as well as the height of those fronds. And we mostly use brush cutting. We decided to try hand pulling on one of the treatment sites, and that didn't seem to make any difference at all. So in terms of data collected, well, we went to collect our data over a period of about a year, and we had our quadrant that we threw down and we counted the number of fronds and the frond height, and then determined the percentage reduction with that little formula. So the teams and working on fire did all the data collection, so I was quite grateful for that. But when it came to the statistical analysis, they just handed me the data. And of course, you can probably see I haven't been at school for a long time, so I had to practice my maths before I arrived at doing a non-parametric test and the Kruskal-Wallis. But we didn't get any statistical significance on our results. But the stats did show that stem reduction appeared to work best at the 8-week cutting interval. And that's really just around what is that interval of cutting when you are ensuring that there's depletion of that underground rhizome resource, and how can you balance that out with cost? So there's a suggestion that the 8-week was the way to go. In terms of the number of, the height of the fronds, the 4-week definitely showed the best result, which again is fairly obvious because these things it grows very fast, but at 4 weeks, it's going to be a lot shorter than it is at 8 weeks. But the 4-week is quite expensive because now you've got to get a team going back onto the site at that time. So it all comes down to this balance of cost versus time, and how does one balance that out? And I think, basically, as I've said, it's, that the 8-week seemed to be the best way to go. 12-week, we seem to be getting too much rootstock replenishment. And typically then you don't get the reduction in the number of stems and the plant seems to be able to rejuvenate. So if you're cutting at an interval that's too long, the plant is simply able to replenish its rootstock and regrow without any issues. Just moving on to discussion, and I'll spend a little bit of time here. First of all, what's interesting, as I mentioned earlier, that we do the fire control with working on fire, and we know that fire seems to stimulate the plants to grow. So what we decided, and it wasn't built into the study as such initially, but it seems to be a good idea to send the teams in fairly soon after those plants have started regrowing. So once that frond is up and out, before it gets to a sort of a 30 centimeter height, it's actually a good time to go in, even with just a manual slasher, or even with just your foot, because those fronds, when they're growing out, they're really soft. So it's quite easy to snap the frond off. So even if it's a small clump, and as I'm sure many of you have seen, these bracken seem to form clumps in grasslands. So if you've burnt an area, right, I've got a patch or a clump of bracken, the team can simply walk through there and stand on those fronds, break the fronds off. That immediately sets the plants back quite a lot. And then again, you can start implementing your 8-week control after that. It's good to get in early in the season after the fire. Obviously, local site conditions will affect your optimal frequency of cutting, and that's something we haven't expanded on further. We didn't look at particular slope aspect issues. So, does the bracken grow faster on the south-facing slopes, the north-facing slopes? We didn't go into that. And it obviously may look different at other grassland ecosystems or at different altitudes, perhaps, or in areas of different rainfall. There's also the practical field constraints. So, some of our areas are quite steep, and you've got to be able to ensure that the team can go in there and work. And also what sort of tools. So if you've got an area where you are able to go in and cut with a brush cutter, fantastic, because you can really move through the bracken quite quickly. If it's a small clump, you can do that just with your foot. And interestingly, some of the landowners in and around Durban that we've met with that are trying to look after this particular grassland type, that have had small patches growing in these grasslands, we suggested just, take a walk, but put it in your diary and do it every 6 or 8 weeks. And they get back there and they just step on the bracken fern, and we've gone back and we've seen a remarked reduction in the plants. So the approach is definitely helping. In conclusion, I think there's quite a lot of future research. This study really just touched on a couple of things, and it's helped us as practitioners really who are trying to manage grasslands and just ensure that there's some sort of cost efficiency in what we're doing and where we're sending the teams, because we've got a lot of other invasive plants which are obviously priorities, and which are in many instances bigger problems still, but we don't want to forget about brackens. So there's a lot more research to be done. I don't think biological control is an option, because I know that's something that people often ask. I'm not aware of any biological control that is available for this particular fern. I've spoken to some of the farmers around the province, and only farmers who are looking after natural grassland. So I haven't been to farmers who are doing livestock or that sort of farming. But the guys I've chatted to, one has used a drone that's loaded with herbicide and has gone and done targeted spraying of bracken and came back with quite positive results. I don't know what that costs, so that's an area which could be quite interesting to try and understand what the cost of drone control for bracken is, and I think for them on a big farm, they're in the Eastern area. It was nice, they didn't have huge expanses of bracken, but they could see it was popping up into the natural grass and more and more. So they decided on that option. I'm always a bit cautious with herbicides, so that's why we've not used herbicide in eThekwini, because we've had bad experiences with that before, and it often depends on who is standing behind your, and actually spraying the plants. So if you've got really highly skilled, trained people, it can work, but you've got to be quite careful. And then another farmer up in the Nottingham Road area, they've used just a tractor with a slasher on to go through natural grassland. And again, the response I got back from those in discussion was that was quite positive, because with the regrowth of grass coming to the area, they found that I think they had wildebeest on the property. The wildebeest were coming back and utilizing the grass in the area where they'd been cutting. So that was quite good. But effectively, let me wrap it up. I think the critical thing is we're going to try and keep monitoring in Durban, but also looking at other ways to control bracken. And I hope that was some of interest to you. Thanks very much.
Thank you very much, Errol, for a lovely presentation on finding a possible method or frequency on when actually the bracken can be actually controlled. Unfortunately, there's no time for questions. I'm trying to keep up so I can be on score with Kevin and be on time with our session, so that he's not the only one on time with the session for this conference. So our next speaker is Sibulele Mboniswa. Is Sibulele in the audience? And as she's coming towards us, she'll be presenting on the impacts of Lantana camara invasion on herbaceous vegetation in the Eastern Cape.
Good afternoon, everyone. My name is Mboniswa Sibulele. I'll be presenting the topic that says invasion impacts of Lantana camara on herbaceous vegetation of Kwelera in East London, in the Eastern Cape, in the Communal Rangelands. Yes. And rangelands cover 7% of South Africa's land and are vital for livestock and rural livelihoods. Despite their significance, rangelands are under increasing threat from invasive alien plants, especially Lantana camara, which suppresses native grass diversity, forage, and alters ecosystem structure. This invasion leads to a decline in palatable species, the spread of unpalatable plants, and overall degradation of rangeland quality. Managing invasive species in rangeland is crucial for protecting biodiversity and ensuring sustainable livestock production. This study investigates the impact of Lantana camara on herbaceous diversity, species density, ground covers, as well as species richness. On our experimental layout, we selected 2 communal rangelands at Kwelera. We demarcated 10 meter by 10 meter plots. Within the 10 meter by 10 meter plots, we placed quadrat 3 times, like vertically, both in the corners and also in the center. Within the quadrats, we estimated covers visually, which we estimated for a litter cover, non-graminoids, graminoids, and also bare areas in a range of 1 to 100%. We used Shannon Diversity Index and also a two-way analysis of variance using general linear model procedure. the graphs showing the invasion impact of Lantana camara on species richness, we can see that non-graminoids cover declines significantly with invasion. Diversity also decreased with an increase in invasion. Table 1 shows the mean percentages of the table shows the percentage means of Lantana camara invaded areas across different invasion levels. Interaction between size and invasion level was not significant, but independently, graminoids cover varied significantly with an increase in invasion. Total density decreased significantly with invasion. Non-graminoids density followed the similar declining trend. To conclude, Lantana camara invasion threatens herbaceous diversity and plant densities. There is an urgent need for restoration interventions to recover herbaceous vegetation and to improve forage productivity in Lantana camara invaded rangelands. However, we recommend future research on soil seed banks to understand a potential for herbaceous recovery and reinvasion. Thank you.
Thank you. I would let you go because you are in a rush also to get off from the stage. Thank you very much, Mboniswa, for your presentation. And then our next presenter for the session is Silungile Dlamini. And while she's making her way to the stage, she'll be presenting on the seasonal use of Pteridium hysterophorus by domesticated goats in KZN.
Thank you. Good day, everyone. My name is Silungile Dlamini, and today I'll be presenting some preliminary results on a study of seasonal use of Pteridium hysterophorus by goats in northern KwaZulu-Natal. Communal rangelands are an essential resource in many parts of the world. They serve as foraging sites for livestock. However, the health of rangelands is under threat from various factors such as land degradation, overgrazing, and invasion by alien invasive plants. The presence of alien invasive plants in rangelands, which are often undesirable to livestock. Oh, sorry, I forgot. Sorry. Oh, so I was there. So the presence of invasive plants in communal rangelands, which are often undesirable to livestock, lowers the grazing value of the rangelands by reducing forage available to livestock, in turn reducing livestock production in the rangelands. Another issue with invasive plants is that they outcompete native vegetation, therefore increasing biodiversity in the rangelands. One such invasive plant is Pteridium hysterophorus, which has invaded over 3 provinces in South Africa, with the province of KwaZulu-Natal being the most affected. Fun fact, it was also first recorded in KwaZulu-Natal Enanda. So KZN has the highest number of households that depend upon agricultural activities for livelihood. So this study aimed to look at the feeding behavior of goats in Pteridium invaded communal rangelands by investigating the abundance of Pteridium as well as determining the consumption of Pteridium by goats. So we conducted this study in 2 villages, Madwaleni and Manzaneni communal areas in northern KZN. Both these areas are categorized as being semi-arid and they highly depend on livestock for farming. For this study, we collected data across 3 seasons, the dry season, the early wet season, and the late wet season.
For objective 1, we used the quadrate sampling method to assess the quantity of Pterodium. And for objective 2, we used direct observations to assess the consumption of Pterodium by goats. This was done by following the goats when they leave the kraal in the morning to the rangelands, and then observing what they eat and how much they eat, and then doing the same thing in the afternoon when they are back around the homesteads. So our results showed that Pterodium was available across all 3 seasons. Across all 3 seasons, more so in the early wet season, but we also found that it was more abundant around the homesteads and the roadsides than in the actual grazing, in the actual rangelands where goats forage. Around the homestead, we found that it was imitating the growth of the grasses in some areas. So around the homesteads, grasses usually grow horizontally. Some of the Pterodium was also growing horizontally in the homesteads. When looking at the actual foraging behavior of goats, we found that they split their foraging into 2. They spend 3 to 6 hours foraging in the rangelands and 2 to 3 hours foraging around the homesteads. So the rangeland is where there was less Pterodium, and the homesteads is where there was more Pterodium. So they are exposed to less Pterodium in the rangelands and exposed to more Pterodium around the homesteads. We found that grasses were the main contributor to the overall diet of the goats across all seasons. However, there was a huge decline in the contribution of grasses to the diet of the goats in the dry season for obvious reasons. When we look at Pterodium and its consumption by goats, we found that it was used across all seasons. Less so in the early wet season, it only contributed less than 3% to the overall diet of the goats. But when we looked at in the dry season, we found that it was the 3rd most used plant in one of our villages. It contributed 15% to the overall diet of the goats in the dry season, and that is equivalent to 4.87 grams per minute. There are various factors that could have influenced this. First of all, when you look at the dry season, you can see that goats feed less in the dry season. They spend almost the same amount of hours feeding in the rangelands where there is less hours, again feeding in the homesteads where there is more Pterodium, so they can't escape the Pterodium. And in the dry season, there's scarcity of nutritious forage for goats. So they may be using Pterodium to supplement for the lack of forage, and also they don't have enough time to look for other nutritious feed around them. So in conclusion, the availability of Pterodium across all seasons increases chances of consumption by goats, whether it is accidental or intentional. The fact is goats are already incorporating Pterodium into their overall diet, more so in the dry season. Unfortunately, Pterodium is also spreading at an alarming rate in northern KZN, and it doesn't seem to be stopping anytime soon. So the higher the plant spreads, the higher are chances of it being consumed by goats more. And the implications of that is that the plant may displace native vegetation as well as grasses, which will lead to less forage available to goats, which will lead to poor animal health for goats, and poor animal health will mean loss of economic loss for the small scale farmers and the landowners and livestock owners in KZN, which will also affect their food security. Thank you.
Thank you very much, Silungile. We are a bit tight now on time, but we'll see if we can catch up. Our next speaker is Yondela Masande Norman. And while Yondela is making his way to the podium, his talk is on determining the effects of restoration interventions on stube, vulgaris, encroachment in the grasslands and in savannas. Oh, and can I please remind our speakers to please keep to the time. If you look through there, there is the timer counting how many minutes you have left.
No pressure. Right, good afternoon, everyone. My name is Yondela Masande Norman, and for my PhD study, I will be doing a speed presentation on determining the effects of restoration interventions on Stevie vulgaris encroachment in savanna and grassland rangelands. So bankrupt bush goes by many names, both common and scientific, but for the intents and purposes of this presentation, I'll just refer to it as bankrupt bush. And its distribution has been well documented. As a member of the Acerosum family, bankrupt bush is indigenous to South Africa. However, due to recent disturbances such as fire, climate change, and changes in land management and grazing regimes, the bankrupt bush has spread to 7 of our 9 provinces here. And that paired with its ecology in that it's a highly adaptable shrub that's able to secrete allelopathic chemicals and compounds into the soil, as well as producing high densities of seed, make it a very formidable competitor in grassland and savanna rangelands, so much so that it ends up taking over the savanna rangelands. Yeah. Anyway, so, which obviously has serious impacts on the rangelands. Besides reducing grazing capacity, it also outcompetes the grass sword as well as the forb species, and in extreme cases, taking over that entire rangeland and turning it into what is known as a green desert. Now, there are multiple control methods that have been used so far to mitigate the spread of bankrupt bush, mainly fire, chemicals, and mechanical removal. But a lot of these treatments have often been conducted in isolation with each other and only with reference to agricultural impacts such as grazing capacity and livestock productivity, whereas the ecological implications of bankrupt bush encroachment and control are still largely limited, which again is the aim of my PhD, which is to determine the ecological and economic implications of the encroachment and subsequent control of bankrupt bush in these rangelands. So our study area is in the Waterberg region, so far here in Limpopo, mainly in what is known as the Ugulima Ranch, which is a commercial game ranch that has been heavily infested with bankrupt bush. And that'll be the site where we conduct our restoration activities. While preparing our restoration plots and experiments, we also took the liberty to go up to our study area and take down some preliminary data using vegetation surveys, soil sampling, and some germination trials, as I'll get to in a moment. So for our first set of preliminary data, we had taken some soil samples from Silungile Sands Nature Reserve, which is within the same region as Ugulima. And we took some soil samples from some bankrupt bush invaded patches in Ugulima. And as you can see here, the soil carbon percentage in Silungile Sands is significantly higher than that in Ugulima. And just for reference sake, Silungile Sands has a far lower density of bankrupt bush. And so the impact of its establishment are far less pronounced compared to at Ugulima. Here we go. We also did some preliminary vegetation surveys while also working out diversity back at Ugulima, where we compared the relatively more natural areas with the heavily encroached areas and areas that we had cleared in 6 by 6 meter plots within Ugulima. And as you can see, the cleared areas are still trying to recover in terms of diversity. The encroached areas have very low diversity index compared to the relatively less encroached and more natural sites of Ugulima. As for our germination trials, we had conducted those at the University of Pretoria's rooftop area, whereby we had taken soil samples from Ugulima and used them as a medium in which to germinate our bankrupt bush seeds. And we have found that so far, over the growing seasons, the average height of the bankrupt bush seedlings has been up to 1.82, and then increasing as we went into the winter season to 2.15 centimeters, suggesting a slow but steady growth. And these will continue to be monitored over time. So the way forward going from here, besides the ongoing monitoring of our sites, We'll also be preparing our actual restoration plots, which should be about a hectare in size each, where we want to employ fire chemical treatments as well as mechanical clearing, and following it up with the reseeding of indigenous grass species as well as forbs. We also want to replicate these assessments that we've done so far, as well as the subsequent restoration activities at a site within the grassland biome, most likely Telperion Nature Reserve in the Mpumalanga Highveld. And furthermore, we also want to, again, not only determine the ecological and economic implications of the restoration of Bangrotbos encroached rangelands, but to also ultimately provide a set of recommendations for landowners that is informed by our data that we've collected on the ground. Right. And that's it. Thank you very much.
Thank you. Thank you very much, Yondela. And our next speaker is Uchwaita. Uchwaita Kabiyana is Uchwaita, okay. While Uchwaita makes the way to the stage, the presentation will be about shrub invasion drives herbaceous vegetation degradation in semi-arid rangelands of the Eastern Cape.
Greetings to the house at large. I go by the name of Uchwaita Kabiyana, and I'll be taking you through a presentation titled Shrub Invasion Drives Herbaceous Vegetation Degradation in Semi-Arid Rangelands of the Eastern Cape Province, South Africa. Shrub invasion is of global concern since it alters ecosystem functioning and services. This phenomenon leads to loss of herbaceous biodiversity and further reduces the forage productivity and grazing capacity of rangelands. Since the 1900s, a dove shrub from the Asteraceae family, Pteronia incana, has invaded vast areas of the grassland and thicket biome of the Eastern Cape. Now, in this study, we aimed to answer the following questions. How does Pteronia incana invasion affect vegetation, herbaceous vegetation attributes and covers? Are the effects of the shrub modulated by slope position along a mountainous landscape? The study was conducted in two communal rangelands of Pedi, namely Lower Mkwalana and Lower Qeto. 3 1-hectare blocks were demarcated at an equidistance of 100 meters in top, middle, and at the bottom slope. Less than 10% cover was characterized as open, between 20 and 40 as moderately invaded, and greater than 40 as highly invaded sites. 3 10 by 10 meter plots were randomly nested in each block, and each plot represents each invasion level. 5 by 5 meter square subplots were marked at the center to estimate the canopy cover and density of Pteronia incana. 0.5 by 0.5 meter square quadrats were placed at the corner and at the opposite corners and at the center to visually estimate the ground covers, which were the graminoids, the non-graminoids, litter cover, bare, as well as cryptogram, and also to identify herbaceous species at species level. We use the Shannon-Winner diversity index to calculate our diversity. We conducted an analysis of core variance using the general linear mixed models to assess the main effects of the slope position and treatment, which was our invasion level, and also their interaction, and shrub cover was used as a core variant on the species density, species diversity, and richness. A post hoc Tukit correction was used to conduct mean separation at 95% interval between our invasion level and our slope position. And we further did a regression analysis. Our results are as follows. The results of the study, we found that the interaction between the slope and the treatment had no significant effect. However, treatment only had a significant effect on the graminoid cover as well as our herbaceous plant density. The open areas of the bottom slope exhibited higher graminoid cover compared to moderately invaded and highly invaded site. And this trend was similar for the top slope. For our plant density, higher plant density was again observed on open areas relative to the moderately invaded and highly invaded. For these two parameters, the slope and the treatment independently had a significant effect, with higher diversity observed at open areas of the top slope. And also for our richness, higher richness was observed on open areas of the top slope. And the top slope had two more species higher in open areas than the bottom slope. We then proceed to our relationships. The relationships indicate that the graminoid cover declined quadratically with an increase in both the shrub cover and shrub density. However, other parameters, they shared a similar relationship where they linearly decreased with an increase in both the shrub cover and density, and other parameters were the plant density, the species diversity, and also the species richness. This study further indicated that higher invasion not only reduced the cover, but also the density and richness, indicating that the shrub facilitates competitive exclusion of grasses. Thus, Terronia incana drives herbaceous plant degradation. Therefore, the findings of the study underscore the critical need for effective intervention, including invasion management and restoration strategies to enhance vegetation recovery. Thank you.
Thank you, Twaika, for enlightening us on how Terronia incana could be problematic in the rangelands. And then our last speaker is Silungile Dlamini, who will be presenting online. So once we have Silungile online, then the floor will be all hers. There we go.
Good day, everyone. I am Silungile Dlamini from the South African National Biodiversity Institute. I am excited to share some fascinating work we are doing to evaluate the potential invasiveness of an emerging alien species, Mahonia oiwacensis. Mahonia oiwacensis is a woody shrub native to China in the Pteridium family. It has naturalized in many regions of the world, such as Australia, New Zealand, some parts of Europe, America, and also in South Africa. It was introduced into South Africa as an ornamental plant. The first naturalized population in South Africa was identified in 2016. It is currently not listed as invasive in both Nemba and Kara. Mahonia oiwacensis is an evergreen shrub or a tree. It has compound leaves and leaflets with spinoserrate margins. It makes beautiful yellow flowers and also these beautiful bluish fruits that are very attractive to birds. Our aim was to evaluate the invasion potential of Mahonia oiwacensis and its eradication feasibility in South Africa, with the objectives to assess its distribution in South Africa, to assess the invasion risk, to evaluate its potential distribution, and also to understand its feasibility of eradication. In our results, we analyzed the records over time since the first record in 2005, and the high R-square value indicates a strong and consistent increase in the records over time, suggesting the species is either spreading, being detected more frequently, or both. Naturalized populations were recorded in Gauteng and KwaZulu-Natal, with one non-spreading population in the Northwest. Populations were recorded in a variety of habitats, including naturalized savanna, forest patches, disturbed areas, roadsides, and riverbanks. The risk analysis and the species distribution model indicate that the species has a significant potential to be invasive in South Africa. For the eradication feasibility, a significant reduction in the number of plants was observed in post control, with smaller error bars indicating that there is a consistent effectiveness of the mechanical and chemical control across all the 3 sites. The seedling emergence was also observed in the 2 sites in Gauteng, but we suspect that this is from the soil seed bank. In closing, we can conclude that the species has a high potential of being invasive in South Africa. However, it is still limited in distribution, meaning that eradication is still possible, although horticultural industry might impede a national eradication of this species in South Africa. We further emphasize that early detection, rapid response, and monitoring are a key to preventing widespread invasion. I thank you.
Thank you very much, Tulisile.